Okay, good afternoon, everybody. Now, I know that today, Monday, you had a test, and normally we don't have class on the days that you have tests, but we're a bit behind. So this week I am putting in two classes as normal, and that will be today, and then we'll complete this topic. You can complete it on Wednesday. You can do these two classes, obviously, whenever it pleases you over the next week. Okay, so we have spent a lot of time talking about, first of all, about genes, about their inheritance. You saw some of the inheritance patterns. And then we talked about the structure of DNA and about how important it was that DNA be replicated accurately every time the cell divides, and especially so that the zygote that results from a fertilization event inherits a complete complement of all of the genes necessary for the cell to survive, but also for it to grow and everything else. So we need then to begin to understand what is the structure and function of the gene itself. What do we mean when we say that a gene is inherited or that a gene has different alleles? In order to understand that, we actually have to understand how DNA functions in the cell. And the central function of DNA relates to the fact that it is a code. It contains a code actually for two classes of molecule. The first, by far the most abundant in the cell, DNA codes for polypeptides. Polypeptides go on to be matured as functional proteins. DNA, however, also codes for RNA. Both of those types of molecules, polypeptide and RNA, can be produced from the code contained in DNA, and that unit, the code that produces a specific molecule, we refer to as a gene. And you will usually hear DNA referred to in terms of its role in producing the proteins inside the body. But remember that it is capable of producing RNA as well. And second of all, in actual fact, the DNA, the gene, when it is initially read off, does not produce a protein. Okay, the whole process ends up producing a polypeptide. It's important to remember that, because a single protein may consist of several polypeptides put together. Second of all, polypeptides need to be matured. Very often they need to pass through the endoplasmic reticulum, then the Golgi, etc., to be modified, etc., before they form a mature protein. So we don't just get a mature protein straight off of the processes that we're going to talk about here. The relationship between DNA and proteins, or rather polypeptides, I'm not going to discuss it. If you want to read about this, there's a very important set of experiments performed, especially by some workers called Beadle and Tatum, and that is in your text, which you are welcome to read. Beadle and Tatum were the first to show that a particular gene led to the production of a particular polypeptide, and that a change in that gene led to a change in the polypeptide or the loss of the polypeptide. Sometimes a change in a gene will mean that it simply can't produce the polypeptide. And that was the work, basically the work of Beadle and Tatum. So the work of Beadle and Tatum and the subsequent workers really has led to the construction of what we refer to as the central dogma. The central dogma states that DNA is transcribed to messenger RNA. It's transcribed to RNA. And the RNA is translated to proteins. And this translation is done by the ribosomes. So that is the central dogma. DNA is transcribed to messenger RNA, which is translated to protein by the ribosomes. Why the central dogma? Well, it's central to all life. All living things use this method, from the prokaryotes on all through to the most complex eukaryotes, basically use this same process. There's very little difference in actual fact between the process in prokaryotes and that in eukaryotes. There's a little bit of difference we'll see in a while. But essentially this same process links all living things because it evolved in the prokaryotes and it has been maintained in all of evolution through everything else. There's nothing living that doesn't use this process, DNA transcription to messenger RNA and its translation to protein by the ribosomes. This just to remind you that DNA codes for two things, for polypeptides and then for RNA as well. You will often hear people mixing these words up, protein and polypeptide, and talking about the one as if it was the other. But in the back of your mind, always bear in mind that a protein is something functional. It's something mature and functional. And it is all proteins are polypeptides, but not all polypeptides are yet functional proteins. Think of it that way. Okay, so we have to remind ourselves now about the structural difference between RNA and DNA. Remembering, DNA is a double-stranded nucleic acid. RNA is fundamentally single-stranded. It may fold back on itself and base pair between different regions of the same RNA molecule, but it is fundamentally single-stranded. Second of all, remember, RNA uses a ribose sugar. DNA uses a deoxyribose sugar. Thirdly, in RNA, there is no thymine. Instead, RNA makes use of a very similar base called uracil. So, where in DNA you would have a T, thymine, in uracil is in RNA. So, for example, if we're base pairing DNA to RNA, we base pair C to G, just as normal. But if I had an A in the DNA, I would match a U in RNA, not a T. So we're going to encounter, in fact, several kinds of RNA. This is the other thing about RNA. There's only one kind of DNA that we need to be concerned with in the cell, but there are several kinds of RNA. The one that we will talk about the most, messenger RNA, but then there is another one which we need to consider, especially here, and that is transfer RNA, and one or two others that you will hear about. One of the things that we will realize about RNA as we're going through all of these processes is that sometimes in the processes of transcription and translation that I'm going to tell you about, RNA itself acts as an enzyme. We refer to it then as being a ribozyme. And it's one of its very, very important functions. So, and I'll mention it each time we see it. So we're going to go through this entire process, DNA transcription first of all, to see how messenger RNA is produced. Then we're going to move the messenger RNA to the ribosomes, and we're going to see how the ribosomes translate into a polypeptide. And then we will briefly discuss some of the ways in which that polypeptide might be matured to a protein. Okay, first, I have emphasized that basically it's the same process for all living things, but there are a couple of differences between prokaryotes and eukaryotes in the mechanics of the process. So, for example, here is a prokaryote, and the DNA is transcribed to messenger RNA, and that messenger RNA is then translated by the ribosomes to produce a polypeptide. This in the prokaryotes, this whole process takes place in the cytoplasm, because that is the definition of the prokaryote. right? It has no organelles. It has no nucleus. The only membrane is the membrane around the outside, around the cell itself. So all of these processes take place inside the cytoplasm, and that has some important implications. One of them being that as soon as messenger RNA appears in cytoplasm, ribosomes will attach to it. So in prokaryotes, what we'll often see is that the messenger RNA may still be being produced by the process of transcription, but it will already have ribosomes on it, translating it and producing polypeptide. That's one important difference. The second thing is here in the eukaryotes, the process of transcription, for example, takes place inside a nucleus, behind a barrier, behind the membrane, behind the nuclear membrane. And the messenger RNA, therefore, has to be exported from the nucleus in eukaryotes, has to be exported to the cytoplasm for the ribosomes to attach. There is a second major difference, and that is that the messenger RNA, the gene itself, is differently structured the way genes are structured in the prokaryotes. And when this messenger RNA transcript is produced, there are some features about it which require that this messenger RNA be processed. All sorts of things have to happen to the messenger RNA before it can be exported. And those processes do not occur in the prokaryotes. You'll hear exactly what they are in a minute. So in the eukaryotes, we don't have this feature of ribosomes immediately grabbing hold of messenger RNA and starting translation. Instead, the entire messenger RNA strand has to be exported to the cytoplasm. Okay, so we're going to consider for a while how this code is constructed, how this DNA code is constructed. Remembering that there are only 4 bases. There are 4 bases in DNA, C, G, A, and T. And there are 4 bases in RNA as well, C, G, A, and U. There's no T, there's a uracil instead of thymine. So that's our first limitation. There are only 4 bases in each of the nucleic acids. There are 20 amino acids that we need to code for. We make all of our polypeptides, so do the prokaryotes, out of 20 amino acids only. So we've got 4 bases, 20 amino acids. Well, we need to realize that the DNA the code is written in words of 3 letters. We refer to those in the DNA, that is referred to as a triplet. So there is a triplet code. 3 bases together compose a triplet. And each triplet codes for something. It codes for, in fact, a corresponding code in messenger RNA. And these very, very importantly, and this is just a simple concept, but it's easy to forget, the triplets lie next to one another. The triplets do not overlap. Okay, they are sequential triplets. They are non-overlapping. And those triplets are actually, first of all, transcribed to RNA, to messenger RNA. Each triplet in DNA corresponds to a codon in messenger RNA. So the triplet is base paired with RNA bases to form a codon, also composed of 3 bases. And each codon codes for something. So here, have a look here. And there are a couple of things to note. First of all, the strand of DNA that contains the genetic information that we think of as being the gene is referred to as the template strand. So there in our example here, here is the template strand, ACC, AAA, CCG, AGT. Okay, so, and there's the template strand. You remember that it is one of 2 DNA strands. So there's a matching strand. Confusingly, it is called the coding strand, but don't get confused. It is not coding for genetic information. It is coding for the template strand. If I destroy the template strand, remember, I can reconstruct it because there is a coding strand, which tells me what the template strand sequence is. That's the first thing. Second, the bases are organized in triplets, ACC, AAA, CCG, AGT. And it is very, very important that in anything that happens here, the cell is able to recognize exactly where to start. I'll tell you why. If I've got these bases, this is a triplet, ACC. Its next triplet is AAA. The processes of the cell need to read that as ACC, AAA, CCG, AGT. It is, if the cell starts at the wrong base, it's going to read completely different triplets. It's going to read CCA. If I started here, CCA, AAC, CGA, that could be a very bad, in fact, a fatal mistake, because the cell would be reading off entirely the wrong code. So the cell the cell processes must begin at a specific place, and they do. There are markers here, as you'll hear about, which tell these enzymes exactly where to latch on, exactly where to start doing their job. So already you realize there's a lot of information contained in this DNA, double DNA strand, right? Templates, that's coding the genetically useful information that we're going to ultimately produce a polypeptide from. The non-template or coding strand, I prefer to call it the non-template strand because it's less confusing. The non-template strand is coding for the template strand, right? It's a matching, but this does not contain useful genetic information. We can't read this off. So for this gene here, shown on this DNA strand, for this gene here, here we have they are saying this is the template strand, this here is the non-template strand. If we move to another gene, it doesn't have to be the same. It could be the other strand. If we move up to the chromosome to an entirely different gene, we may find that this now will become the gene, the template strand. So up here, this may be the template strand, the opposite to this arrangement here. Just realize something. If we're reading the code in this direction on this gene, then if the strands, now the template is on the other strand, it will be read in the opposite direction. Okay, so it's all the gene is read 3' to 5', and we'll see exactly why in a minute. So here we're going to actually transcribe. Remember the central dogma: DNA is transcribed to messenger RNA. The reason for that is the ribosomes do not talk DNA. They are unable to read DNA directly, and they need an intermediary set of molecules in order to be able to interpret the code. So a transcript in RNA is made, and that is what ribosomes will be able to read. And so here it is here. Remember, here's our first triplet, ACC. Here's our first codon, UGG. It's got a match there, ACC, UGG. here's the second triplet, AAA, is going to be U in RNA, so on, so on. GGC, UCA are going to be the matching RNAs. And just a little trick for you. If you want to know what the messenger RNA reads, just look at the non-template strand, which is matching the template strand, right? There it is there, and substitute U for T, and you'll be able to predict what the messenger RNA would be. So this is what is sent out to the cell. This is what the ribosomes can react to, and they are able to read this. But there is another problem, and that is the ribosomes have got to make a strand of polypeptide. The monomers of polypeptides are amino acids. The monomers of all of these things, of DNA, RNA, the monomers of the nucleic acids are nucleotides. How do we get from the nucleotides to a chain of amino acids? Well, what you have to do is you have to rely on an intermediary. It's the same as if I speak French and German, but I don't speak Finnish, right? And I want to have a conversation with somebody who only speaks Finnish and German. What have I got to do? I've got to go find somebody who speaks Finnish and German, who can speak to the Finnish person and speak to me in German, and then we can somehow cobble together our conversation. It's the same in this. The cell needs an intermediary who speaks both nucleic acid and amino acid. And that intermediary is called transfer RNA, a very strange molecule that is mostly nucleic acid, but which can bind to and carry an amino acid, as we'll see in a minute. So the transfer RNA is both nucleic acid and amino acid. And that is that is going to be the link between there and there. The transfer RNA is the key to the process of translation. Don't worry about, we go through it slowly, and if you can't, and you'll see what's happening. And I'll happily repeat. Okay, so there are, let's just say, first of all, I want you must be able to do these as problems as well, and that is take. Let me just say it this way. You must be able to look at these strands. Let's say I just give you the sequence of the template strand. You must then be able to tell me what the messenger RNA strand is, and then eventually be able to tell me what the transfer RNAs are that come in and what amino acids would be put in place. Be prepared also to be given things like, just be given this non-template strand and be asked what amino acids, what polypeptide would be produced from a particular sequence. And it's always done just as a simple matter of matching bases. But then eventually, what we're going to come up against is how do we make a jump from messenger RNA codons to the amino acid sequence itself? And the answer is that when we look at the codons, remembering that there are 3 bases in a codon, there are 64 possible combinations of the 4 RNA bases. Okay? The bases being cytosine, guanine, adenine, and uracil. There are 64 ways of combining those into triplets, into 3 base codons. So we've only got 20 amino acids. Okay, we've got 64 possible codes and 20 amino acids. So in actual fact, a single amino acid may be coded for by several different codons. We say that because of that, the code is redundant. Sometimes you'll see it called degenerate. So an amino acid, a single amino acid, may have several different codons that code for it. That is an important implication, which we'll see in a minute as far as the transfer RNA is concerned. But the code is unambiguous. That means no codon codes for more than one amino acid. Okay. Each codon codes for only one amino acid, but any amino acid may have several different codons. And again, this brings out the point that we must be reading in the correct, start at the correct codon and start at the first base of the correct first codon in order to read the sequence properly. We call that the reading frame. We must be in the correct reading frame in order to read this properly. So there was a lot of work done in early molecular biology to work out what the sequence of each of the codons was. And here they are here. This is the version given in your book, but I have given you here the same table. It's just got all of the names written out in full for you rather than their abbreviations. Okay, so let's have a look here. This is a table. Always remember, this is a table of codons. This is messenger RNA. This is the messenger RNA code, not DNA, not transfer RNA. This is the messenger RNA code. This side here is the 5' side. That's very important. This is the 5' side. So here we have all 64 possible codons, and we have all of our 20 amino acids. And you'll see that all except 3 of the codons, this one, and this one, code for an amino acid. These 3 here, UAA, UAG, and UGA, these 3 code for, it says stop. You'll see their function in a while. There is one other important one, and this is one I don't expect you to remember these. Okay, you will always be given the table of codons to work from in any problem or anything else. But here is the other important one, and it's this one. This one I do want you to remember. This is the codon AUG. So it's 5' AUG. That codes for 2 things. It codes for methionine, which is an amino acid common in proteins. It occurs over and over and over and over again. But the very first methionine in a sequence is the start. Translation always starts at a methionine. And there is a methionine which is marked as being the very first one. And it's at that point that translation begins. Okay, so the second thing to emphasize is this. As we go through our central dogma, the central dogma applies to everything, to all living things. That genetic code that we see is universal all the way from bacteria on up to the eukaryotes. And we can experimentally show this, because if we can extract genes and insert them into a genome, we can insert genes from wildly disparate species. So, for example, here is a cat, and this cat is glowing. It's glowing in the dark, and it's glowing because it has had a gene from jellyfish inserted into its genome, and it's happily expressing it. No other effects on the phenotype whatsoever, but it's expressing jellyfish fluorescent protein. Isn't that extraordinary? It's just an object lesson that this language contained in the DNA code is the same for all living things. So you have the potential. This is the huge potential of genetic engineering, is that you can of recombinant genetic engineering, you can actually move genes from one species to another and have them be expressed. Okay, so we're going to start right at the very beginning. That's all this introduction for you. If you are already lost, stop right here, go back, repeat what I have said, because there's information there that you do need to bear in mind as we carry on. So we're going to go through the whole process. We're going to look at transcription first, and then we're going to go and have a look at the process of translation. So transcription is the very first stage of gene expression. When a gene is read by the cell to produce, ultimately to produce polypeptide, we call that gene expression. And the first stage of gene expression is that we need to produce a messenger RNA transcript from the DNA, from the template strand. And that is the process of transcription. The RNA strand, the messenger RNA strand, as you've seen already, is complementary to the DNA template strand. So it matches base for base, excepting that where there is an A, you don't put in a T in RNA, you put in a uracil, U. In some ways, you'll see some similarity to the process of replication. And the difference is that what we're actually producing is an entire RNA strand, messenger RNA, and the transcription just begins without having to lay down primer or anything else. But other than that, the base pairing rules, etc., are the same. This is just an overview. This is not our final go-round. So I just want to show you. Here's our strand of DNA. This is the gene, or the transcription unit. Very often, the messenger RNA encloses the genetic information, which we would regard as being the useful part of the gene, in other pieces of DNA. So this may be longer than the actual gene itself. So the process begins, we're upstream of the gene. It begins with the attachment of a whole set of enzymes and proteins, including an RNA polymerase. And here's our RNA polymerase. And what it does is it unwinds the DNA for a little bit, and it lays down the first RNA. Now, I want you to realize why I'm always emphasizing that you need to look at the priming. Here is the gene. Here's the template strand here. And it's really important. There's its 3' end, there's its 5' end. The RNA polymerase is going to lay down a strand, and it is going to grow in this direction here. It's going to grow in that direction. Why? Because this is the 5' end of the RNA. And just like any nucleic acid, the only place we can add bases to this messenger RNA transcript is at the 3' end there. That constrains it. It has to be going in this direction there. It has to be using this as the template. I remember what I told you. For another gene, it may be quite different. For another gene, it may be this strand that is the template. But in that case, it would have to run in that direction, okay? Which it can do. That's common all over the cell. We have genes that are in opposite directions like that. This process of transcription, it takes place relatively slowly compared, say, to how rapidly replication takes place. But it carries on. It produces this is the RNA transcript, carries on and on and on, until it reaches an area where it is instructed to stop. There is not necessarily a single codon there that says stop. There are various ways, depending whether you're talking about prokaryotes or eukaryotes, and several in different ways in prokaryotes, depending on the gene. But somewhere down here, this whole complex falls off the DNA, and the transcript is liberated. In prokaryotes, they would already be using it to translate. In eukaryotes, it's still got a little bit of processing to go before ribosomes can get to it. Okay, so the first thing to realize is that the RNA polymerase has to attach to the DNA, and it does so in a region upstream called the promoter. And there are some important little sequences in there, which tell the various proteins and enzymes, this is where you need to attach. That promoter region can act as a switch. The cell can actually switch a gene on by attaching what are called transcription factors around the promoter. And if they attach the correct transcription factors, then the RNA polymerase can come in and fix right there and begin the process of transcription. The whole area that is transcribed is called a transcription unit, but realize that it may have additional messenger RNA on either side of the useful RNA that is going to be translated later on. So there are three stages: it's initiation, then elongation, and finally termination. So the promoter is really important in control of gene expression, because it's there that we can actually operate almost as a switch. And sets of proteins are brought in to bind to the promoter, which are called the transcription factors. The transcription factors actually guide the binding of the RNA polymerase. And if the transcription factors are not present and correct, the RNA polymerase can't bind tightly enough to begin the process of transcription. There is one really important area of the promoter in eukaryotes, which is called the TATA box. It's actually a mixture of T's, thymine, and adenines, TA. This is a critical area where the RNA polymerase will actually come in and bind tightly to the template strand. So here it is here. Here's the TATA box there. The TATA box actually encourages the binding of transcription factors. And the transcription factors are kind of placed so that they encourage the RNA polymerase to bind tightly to the DNA. So here it is here. And this we refer to as the transcription initiation complex. So the whole thing eventually is going to be, we'll refer to it as the transcription complex. But the critical part of the complex is this, which is the RNA polymerase. And the RNA polymerase is able to separate a small area of the DNA bases, one from the other, like this, and immediately begin laying down an RNA strand, which is going to form our messenger RNA strand. And as I told you, it's going to form in this direction this way, because we have to add at the 3' end there. So it unwinds about 10 to 20 nucleotides at a time, not very much, not nearly as much as in replication. But it also moves relatively slowly, about 14 nucleotides per second, as opposed to the hundreds or thousands that are replicated during DNA replication. As this RNA polymerase moves along, another RNA polymerase can latch on behind. You can produce several messenger RNAs if need be. You can carry on producing messenger RNAs as long as the cell needs the product of the messenger RNA. So here it is here, the growth of the messenger RNA strand. It's quite simple. It obeys the same rules, and it's done in much the same way as a new DNA strand would be made during replication. These come in as triphosphates, and the phosphate bond is broken, and a phosphodiester bond is made to form a new nucleic acid chain, and in this case, it's RNA. This little hybrid area, RNA, DNA, doesn't persist. Instead, the RNA is liberated from the DNA, and the DNA bases match up again behind the RNA polymerase. And it just moves all the way along the DNA strand, reading until it reaches a point where there is a signal to the RNA polymerase that it needs to disengage. And there are several of these. There in eukaryotes, very, very often, there is a TA signal, or TTTT, whatever, that signals to the RNA polymerase that it should loosen and fall off. And it does so several bases past a signal that reads TTT or TA. And I won't go into the details, but in prokaryotes, there are various ways in which the DNA actually kicks the RNA polymerase off. So we're left then, at the end of transcription, we're left with a length of RNA, which we refer to as being messenger RNA. It's messenger RNA. It has been read directly from the codons of the DNA strand. And in prokaryotes, it's fully functional. Immediately it's produced. In eukaryotes, it's not. In eukaryotes, that messenger RNA needs to actually be processed. And processing takes place inside the nucleus. And the first thing that happens is the ends of the messenger RNA molecule are altered. They have things added to them. And the first thing is there is a cap added to the 5' end, and then there's a long chain of adenines added to the other end. And it's difficult to work out exactly why this has to happen, but it seems to largely have to do with marking this messenger RNA for translation. There are many RNA molecules present in the cell, and this may be a way of identifying this as being special, as being messenger RNA, which needs to be translated. It also appears to protect the RNA for some time from being chopped up by enzymes, which are designed to break down RNA inside the cell, because RNA is often injected by viruses. The caps seem to assist in binding of the ribosomes. So here's what the messenger RNA looks like in a eukaryote. This here, there is the poly A tail, lots of A's together. That there was the signal in DNA. It would have read TTA TTT in the DNA on the coding on the template strand. That's the polyadenylation signal, which told the RNA polymerase get ready to fall off and stop transcription. Have a look at this end, and you see there is a cap. The 5' cap here is guanosine triphosphate has been added. This area here is a length of the messenger RNA which will not be translated. It's called the untranslated region, the 5' UTR. And included in this is information for the ribosome. And one of the important pieces of information that it contains is a little sequence called the Shine-Delgado sequence, so which I'll tell you about. And the Shine-Delgado allows the ribosome to begin translating at the correct spot. We'll see exactly why. This here is called the start codon. Remember, I told you methionine also functions as a start. Here it is here. The first methionine after the Shine-Delgado is where translation will begin. Now let's have a look here at this along here. The part that is going to be translated will start here and it will end at one of our stop codons there. But in between, in eukaryotes, not in prokaryotes, but in eukaryotes, there are big areas which do not code for any polypeptide. And this is very strange. Here they are here. They're called the introns. Introns because they intersperse in the whole sequence. The bits that will be translated are called the exons, and here they are here. So before this messenger RNA goes out from the eukaryote nucleus to the cytoplasm to be translated, the introns are clipped out and the exons all joined together. This is done by a special group of RNA and protein enzymes called the spliceosome. And the spliceosome takes out the introns, clips all the exons together. And you can remember exon because these are the regions which are expressed in the cell. These are the regions which are going to be translated. So have a look here and you'll see, here's the intron there. Here's another big intron there. Here the exon there, there, and there. And so before this goes out to the cell, these introns are clipped out and the exons are joined together. The exact function of the introns is not always clear. They definitely do have functions. They seem to have important information contained in them, but exactly what all the function of the introns is not really known. We do know that very often, for example, a protein which a polypeptide may fold itself, for example, into complex regions, and there will be distinct domains of the polypeptide where it will fold itself. And these exons often, the exons, sorry, there, and there, the exons often correspond to different regions of the same polypeptide, to different domains of the polypeptide and the eventual protein. Okay, this just tells you about spliceosomes. All right, so now we've got our transcript. In eukaryotes, it's sent out in this form here to the cytoplasm, and now the ribosomes need to actually attach to it, and they need to be able to read it, and they need to be able to read in the correct reading frame. They need to start at a specific place. They need to end at a specific place. And in between, they need to remain in the correct reading frame so that they read the correct sequence, codon by codon by codon, to produce a polypeptide, amino acid by amino acid by amino acid. And in order to do so, we have to have a look at this other kind of RNA, which serves as an intermediary. It acts as an intermediary between the ribosome and the messenger RNA. So both the messenger RNA and ribosomal, and the ribosome itself are composed of nucleic acids. The ribosome itself is composed of ribosomal RNA, which has two functions. The first is structural. It is folded into the shape of the ribosome. And it is accompanied by specific proteins in the ribosome. But the second thing about the ribosomes is that their RNA has enzymatic function. It is also ribozyme. So a lot of the reactions that take place in translation are catalyzed by the RNA of the ribosome itself. And so we have the ribosomal RNA talking to the messenger RNA. But in order to translate the messenger RNA code to a chain as a sequence of amino acids, we need to have this intermediary molecule, which has amino acid associated with nucleic acid. And that is this incredible molecule called transfer RNA. So let's just have a look at an overview of the whole process. Here's our messenger RNA strand, single-stranded, the 5' end here, 3' end there, organized into 3 base units, each of them a codon. This is a ribosome. Ribosomes consist of 2 halves. There is a small portion here, and there is a large portion here. And they sit on the messenger RNA in a very strict orientation. Inside the ribosome, there are 3 different places, 3 different sites. They're called the A site here, the P site here, and this one here is called the E site. This is A, you can remember, it's in the amino site. This is where the amino acids are going to be brought in. The P site, polypeptide site, that's where you'll most often see the chain of polypeptide. The E site is the exit site. So what is going to happen is here we have the ribosome sitting on the messenger RNA in a particular orientation. Here are the codons, 5' U, G, G, 5' U, 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 5' G, G, C. We're going to make use of this other molecule, transfer RNA. And transfer RNA has bases at the bottom here, which will match to the bases in the codon. So here we have 5' G, G, C. In the transfer RNA, we would have C, C, G to bond there. And this tRNA carries a specific amino acid. And that, in this case, the amino acid is glycine there. And if you go to that table of codons and you look up the mRNA codon GGC, you will see that that is the codon for glycine. That codon can only code for glycine, can't code for anything else. And the tRNA matches it with these 3 bases here, which we call an anticodon. So here is the basic process. The ribosome is going to run along this messenger RNA, and it's going to repeatedly pull in tRNAs, one after the other, which match to the whatever codon is in this A site. Okay. In doing so, we're going to end up making a chain of amino acids, a polypeptide, in a specific order determined by the order of the codons, which in turn was determined by the order of the triplets in DNA. So let's have a look at a transfer RNA. We will seldom see it in its more complex form. We usually just simplify it like this. A tRNA is a relatively short molecule. It's about 80 bases long. It's folded in space in a 3-dimensional shape. When you flatten it out like this, is sort of a clover leaf, but this is more realistic here. It's like an upside down L shape like that. Remember, it's a single-stranded RNA molecule. There it goes all the way around, 3' end there, 5' end there. But it can base pair to itself. And so C to G, U to A, etc. And you'll see there are 1, 2, 3, 4 areas which will actually form helices in space. And they twist the molecule and into this upside down L shape like this. There are 2 areas of the molecule you need to pay a particular attention to. The one is the 3' end, because the 3' end can attach an amino acid. And then you also need to pay attention to this area here. There, those 3 bases there are what will match to the codon of messenger RNA. So this is called the anticodon, 3' AAG in this case. Okay, 3' AAG in this case. So here's our molecule that can talk both nucleic acid and it can talk amino acid, because it's a bit of both. The amino acid is joined here by a very specialized set of enzymes called the amino acyl transferases. And the amino acyl transferases correctly attach the correct amino acid that corresponds to this anticodon. Okay, so the full name is amino acyl tRNA transferases. And they are responsible for charging the tRNAs. And they charge them with the correct amino acids. Very, very important that that happens. So, by the way, we're going to take that amino acid, join it to a polypeptide. These tRNAs are recycled the whole time. They bring in amino acids, they go back, they get recharged, they come back again. We were going to represent them quite simply, just with this little symbol here. But what's important is here is their anticodon, and this is the amino acid attachment site there that we're going to have to watch. So the one thing that I do need to tell you, we don't actually have to have 64 or rather 61 different tRNAs. One tRNA can in fact match to more than one code, provided it's for the same amino acid. And I'll show you why. Let's just have a look here at this amino acid here. This is proline. So these are the codons, CCU, CCC, CCA, CCG. tRNAs, as long as they read their anticodon, read 3' GG, doesn't much matter to them which codon, which base follows. There are a couple of different tRNAs reading GG something, can carry proline, because they're not going to be able to match to anything else. There's nowhere else where the anticodon would read GG. So they're a little bit promiscuous about this last base, and we call that wobble. We say that they're able to wobble because they don't have to bind tightly here to this last one. It's these two that are important. So GG on the anticodon would allow them to bind to any of these. It means the cell doesn't have to have as many different kinds of tRNA. But notice that no tRNA can code for 2 different amino acids. And many of the amino acids, in fact, do have a couple of different tRNAs that will code from. Let's have a careful look again at this table of codons, and let's just see how it actually works. First of all, remembering that what we're looking at is the messenger RNA sequence put together into 3 bases at a time as a codon. If I ask you, if I say to you, well, what amino acid does GAU code for? GAU. Well, here's how to use the table. You can pick through and find it if you want to. but it's much quicker. We're going to say GAU. The first letter is G. It is somewhere in this box here. Okay, that's the first letter. Here's the first base, G, somewhere around here. The second base is A, so it's here. It's in here, where this A intersects with this here. And then the last letter is U, and there it is there. GAU, it's a codon for aspartic acid, aspartic acid. We can work it out for the anticodon as well. Let's have a look here. Here, we said here's the anticodon, CCG, right? So what CCG, just remember CCG, what is it? What is the messenger RNA that is going to match to? It's going to be GGC. Let's see. GGC. There it is there. And there it is, glycine. That's exactly what we saw that tRNA carrying, right? GGC, that is the glycine. And the anticodon that's on that tRNA, go back and check again, CCG, to base pair there and bring in glycine where this codon occurs. So that is basically the way in which it's going to work. The cell itself interprets those codons. So first of all, I'll remind you, there are 64 codons. There are only 20 amino acids, and 3 of the codons do not code for an amino acid. That is these 3, UAG, UAA, and UGA. Those are stop codons, and that is an instruction, stop. And when that codon occurs, translation itself ends, and the ribosome falls off and stops doing its translation. Methionine, AUG, is a start codon, and we'll see now exactly how that works during the process of translation. So here, have a look here, and just to remind you about the structure of the ribosome. We have a small subunit there, and then we have a large subunit here. The large subunit has these 3 sites, this there, A, the P site, and an E site. At A and P, there is actual bonding between transfer RNA and messenger RNA. The A site is, the full name is the amino acyl tRNA binding site. Just remember A, amino, and you'll remember. The P site, the proper name is the peptidyl tRNA binding site. Just remember the peptide site, and you'll be able to keep those in your mind. Okay, the ribosome needs to match to the messenger RNA at the correct place, and it needs to start translation at exactly the correct place. And that is done by a simple set of rules that have to be obeyed immediately at the start of translation. So we're going to see initiation in translation is incredibly important, so that the translation proceeds in the correct reading frame. So we have an initiation, and we have elongation as we had before. We're going to see our amino acids all joined together to a polypeptide chain that gets longer and longer. And then this is going to end. It's going to be terminated when the ribosome encounters a stop codon. At each stage of the game, ribosomal RNA is performing many of the catalytic functions that are required, but there are also other proteins, other enzymes that come in, which we won't talk about in any detail. This is also an energetic process, so it does require the input of biologically useful energy, usually in the form of phosphate bonds attached to nucleotides, as we have seen elsewhere in the cell. But in this case, very often GTP, guanosine triphosphate that is used rather than adenine ATP. Okay, so let's have a look first at the process of initiation. So here's our messenger RNA strand. It has been produced by the process of transcription. It's arrived in the cytoplasm. If it was eukaryote, it had all sorts of processing that took place. Upstream of the start site, there is a little sequence called the Shine-Delgado sequence. That sequence is important because the small ribosomal unit initially binds there. And when it binds successfully to the messenger RNA, it will move down the messenger RNA until it encounters an AUG, a codon for methionine. So it's up here is this little sequence, the Shine-Delgado. The ribosomal portion moves down, subunit rather moves down to this first AUG, and that is the start signal. It's also, of course, the codon for methionine, and the small ribosomal subunit will bring in a tRNA, and here it is here. And if this is AUG, then the anticodon must read UAC, 5 from AUG, 3, and so the tRNA is UAC. And here it is here with methionine attached to it. That has been attached by the amino acyl tRNA synthases. So this small subunit is very important because it does a lot of work at the beginning of translation, and it checks to see that all of this is correct. It checks to see that this tRNA is correctly aligned, AUG. It checks to see that this tRNA carries the correct amino acid, etc., etc. If indeed everything is correct, with the assistance of some other enzymes, it brings in the large ribosomal subunit here, and that large ribosomal subunit orients itself specifically so that this here is in the P site, and there it sits there. So that's the initiation complex. Small subunit, large subunit. Here's the start codon, and here's the first tRNA with methionine there. And now our translation can begin in earnest. So translation is a very complex process, but I'm going to show you this, and I'm going to give you a couple of rules, which if you can remember these rules, the whole process will become quite clear to you and quite easy for you to remember. Okay, first of all, get clear in your mind the orientation of the ribosome: A site, P site, E site. Okay, here's the messenger RNA here with its codons, and here comes the tRNAs with their anticodons and matching amino acids. The rules are that an empty A site must immediately be filled. And it is usually filled with by bringing in a tRNA which matches its anticodon to this codon. There is an exception to that, and that is when we finish. When you hit a stop codon, something different happens. But while translation is proceeding, as soon as there is an empty A site with a naked codon at the bottom, the cell brings in a tRNA to match that together with its correct amino acid. So an empty A site must always be filled. That's rule number 1. Rule number 2 is very important to remember. When we create the amino acids, the polypeptide chain, we take this entire chain here, which is at the P site, and we join it there. Okay, this goes there, not this way. We don't take this and add it there, or any other combination. We take this entire chain and we join it here with a peptide bond. That is the responsibility of the RNA contained in the ribosome, to direct that process. And this forms a peptide bond between this chain, whatever it is, and the new amino acid. So this new amino acid is always the end one. It's always the first here, attached to its tRNA still, okay? So, fine. No empty A site, bring in tRNA. As soon as the tRNA comes, this chain is joined to the new amino acid. Here's the next rule. An uncharged tRNA is not tolerated and has to be evicted from the ribosome. And the way that that is done is that the whole ribosome moves relative to the messenger RNA, and it moves one codon. Just picture it in your mind. Even if my description is difficult, just look at this and imagine this ribosome moving in one codon in that direction. One codon in that direction would put an empty tRNA in this site here. It would move whatever was in the A site to this site here, right? What won't the cell tolerate? It won't tolerate an empty A site. It's got to bring in another tRNA, right? As soon as that tRNA arrives, this chain will be joined to it by a peptide bond, leaving this empty. The cell won't tolerate that. As soon as there's an empty tRNA here in the P site, the ribosome moves, we call it translocation. It translocates one codon to put the empty tRNA in this E site. It's kicked out. Now we've got tRNA sitting with a chain, but we've then got another empty A site. Another tRNA comes in, and the tRNA has to match the new codon together with its attendant amino acid, and so on, and so on, and so on. This can go on for many thousands of amino acids may be joined together in a peptide chain in this process. And eventually, we'll see how it ends that. So let's do elongate with initiation, with binding, finding the Shine-Delgado, moving to the start codon, bringing in the first tRNA, and then bringing in the large ribosomal subunit. Once that had happened, then there was an empty A site. The next tRNA was brought in. By the way, these empty tRNAs, again, I repeat, they get recycled. They immediately get recharged with their appropriate amino acid, and they can participate again. So this process happens quite fast. In bacteria, 600 to 1,000 amino acids in a minute can be added to the chain. This just summarizes for you the whole process for you. So here, for example, we have a tRNA. This ribosome is just translocated, so it's kicked out an empty tRNA, and here sits a tRNA in the P site together with a chain. Cell won't tolerate an empty A site. It brings in a matching tRNA here. Immediately that happens, this chain joins to the new amino acid, leaving this empty. It won't tolerate an uncharged tRNA in the P site. The entire ribosome moves one codon, translates one codon to place this empty tRNA in the E site and evicts it. And then we're back to here. We're back to having now a chain in the P site, empty A site, bring in the new one, etc., 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 on and on and on and on for as long as this series of codons says bring in an amino acid, bring in an amino acid, bring in an amino acid. But eventually, the placed in that sequence will be a stop codon, UAA, UAG, or UGA. And as soon as there is a stop codon is encountered at the A site, as soon as this translocation exposes a stop codon, there is no transfer RNA. There's no corresponding transfer RNA that will fit in there. Instead, a set of proteins are brought in, which are called the termination factors. And they cause the ribosome to actually disengage from the messenger RNA, fall off, and they cause this polypeptide chain to be liberated as a free polypeptide. So that stop codon signals for the release termination. It is a real stop. Translation stops dead as soon as a stop codon is encountered at the A site. So that's here, you can see here, here's the stop codon, and here are the releasing factors brought in place of a tRNA. And they actually cause the hydrolysis of this bond here between this last tRNA and this long peptide chain. They cause this to be hydrolyzed, released, and the whole thing falls apart. Remember, there may be several ribosomes reading this messenger RNA. It's not necessarily the end of the messenger RNA. The messenger RNA does have a limited lifespan in the cell. You don't want the messenger RNA to be hanging around being translated long after the need for the product has been exhausted. It's just a waste of energy. So all the messenger RNAs have a limited lifespan inside the cell. They are eventually degraded. Doesn't matter. If you need more, read it off the gene and carry on producing whatever it was, the product that you needed. Okay, so I'm going to leave that there for today, and I'll complete this at our next class again. This week there will be 2 classes, okay?